Welcome back, everybody. So today we're going to transition from digestion to metabolism. And specifically, we're going to talk about how the nutrients that were absorbed during digestion are now interconverted to either produce energy or to produce molecular building blocks for the body by various cells and organs in the body. And so in today's lecture, I'll both be doing the transition, relating a lot of concepts from metabolism or that are important for metabolism to those that we covered in the digestive system, but also really breaking down the principles of metabolism in terms of what we're looking at, what processes are important, and what macronutrients we're really going to be concerned about. And ultimately, all of this is going to come down to the nutrient pool. So our macromolecules here, our proteins, our carbohydrates, and our lipids, and how those can be interconverted between each other, and then also broken down to produce energy in the form of ATP. And that process, as a reminder, is cellular respiration. In the digestive system unit, we talked about how molecules were broken down from large macromolecules, in this case, a polysaccharide or a protein, through the process of both mechanical breakdown, so things that unfolded or broke down interactions that weren't actually atomic bonds, so for instance, hydrogen bonds, which are charged interactions, and then chemically digested. And remember, that process of chemical digestion involved the actual cleaving of atomic bonds and breaking down our molecules. And so we went from a large polymer, which is composed of multiple different monomers, or the smallest unit of that specific nutrient or organic molecule. And so we take that large polymer, and through this series of mechanical breakdown and chemical digestion processes, we end up with these monomers, which were then small enough for the body to absorb in the small intestine primarily. And that was driven by various charge interactions, the size of those particles, and also the properties of the alimentary canal itself in terms of surface area for absorption. And so we talked about a lot of these different things, and also how different organic molecules were absorbed. So for instance, carbohydrates were actively transported, which is also true of amino acids, but then lipids could freely diffuse through the membrane. And so there were differences in how things could get inside the cell. Specifically, and related to metabolism, we talked about how in order for things to get into the body in the small intestine, they have to be effectively monomers or dimers, but pretty much monomers. So you need to break your carbohydrate, your complex sugar, down to the individual sugar molecules in order for them to be small enough for the body to absorb. So a lot of the processes in the digestive system, or pretty much all of the chemical digestion processes, are best termed as catabolism, which is the breaking down of larger molecules into smaller components, and then these smaller components get into the body. And there were lots of specializations for the small intestine. And so now we're going to move into what happens to these once they're inside the body, because now the body has these little building blocks. You can kind of think of them as like chemical Legos, and then or Lego, and the body will take those and create larger molecules or also break them down for energy. Before we get to that, I want to just quickly review some of the accessory organs, and we'll focus on a specific one, the liver, and go a little bit more into its functions beyond simple digestion. So remember, the pancreas had the function of producing these water-soluble enzymes, which actually, represented by these scissors here, which actually broke the atomic bonds and catalyzed those reactions, allowing those to happen to produce these monomers. And so we'll see a lot of similar processes when we talk about metabolism, but instead of monomers, we'll be looking at how these monomers are broken down into even smaller components, specifically carbon dioxide and water, or we'll look at the reverse of this process of breaking down things, which is building them up, which would be anabolism. But again, the same basic ideas will apply. You need these enzymes to catalyze these reactions and allow interconversion. In addition, we looked at the function of the liver and the gallbladder for producing these bile salts, which were involved in mechanical breakdown. So basically turning oil so that it could be broken into smaller particles in water via the production of these micelles, and then small enough to absorb in the gut. However, the liver is much more important than simply the organ that produces bile. The liver's primary function is actually in metabolism, and it is the most important metabolic organ in the body. It does almost all of the complex biochemistry in the body. And so if you'll remember, we talked about kind of this liver structure of these lobules and how blood flows through it. And we have these hepatocytes, which are the liver cells. And you'll have hopefully recall from the dissections of the mink that the liver is just one solid mass. It's very solid because it's almost completely composed of cells and blood vessels. And each of these cells is a little biochemistry factory. And to illustrate its importance in this metabolic process, we can simply look to blood flow from the digestive tract. So as a reminder, all the blood, or most of the blood from the digestive tract, flows back into general circulation via this hepatic portal system. And so this is composed of multiple different veins, but here we have them color-coded and described. And so here we have the large intestine, the colon right here, the small intestine, the stomach, the pancreas, the spleen, and you'll note that all of these blood vessels are collecting to form this hepatic portal vein. As a reminder, we have had a bunch of capillaries here, so the arteries came in, supplied blood through the capillaries to the digestive organs. The nutrients are absorbed in those capillaries and lymphatic vessels, and then they flow and they drain into the liver via this hepatic portal vein. And so this hepatic portal vein then feeds into these lobules of the liver, and all of these nutrients flow past these hepatocytes, where they will be in high concentration in the blood and diffuse down their concentration gradients into the hepatocytes, which can then perform biochemical reactions. And then once they're done with it, that will all drain into this central vein right here, and then that will then take the blood eventually to systemic circulation in the heart to be recirculated to the rest of the body with the absorbed nutrients after they've been processed by the liver. And so this is really the key thing. The liver is mainly a metabolic organ, although it does play a role in digestion in the form of producing bile. So I like to think of the liver as kind of chemical digestion plus, because it doesn't just break things down, it's really important for building things up. And so these hepatocytes have a lot more enzymes than even the pancreas secreted into the small intestine. So the liver is really a biochemical superhero in terms of the body. And so all of this chemical interconversion is occurring in the body proper. So we're no longer in the alimentary canal in that lumen. We're actually looking at what happens to the nutrients once they're inside the body, in circulation, and they're already microscopic and small enough to flow through the blood. Just to illustrate this idea of metabolism, in a kind of a different version, because the liver is so important, but it really gets short shrift in the A&P because we just don't have enough time to talk about it. I wanted to talk a little bit about its role in detoxification and use the conversion of Tylenol or metabolism of Tylenol or acetaminophen as an example of this. So normally what happens when somebody takes Tylenol or acetaminophen is it's metabolized immediately by the liver. So note that when things are absorbed, they go into the small intestine, including any medications you've taken orally, and before they get to the rest of the body, they actually go to the liver. And so the liver actually gets the first pass at everything that is absorbed, including drugs, and the liver will often interconvert these into different chemical forms before they ever get to circulation. And so this is a really important consideration in drug design, because often if you're giving an oral drug, whatever drug you give the person, you have to assume it goes through the liver first, at which point the liver will do some biochemical reactions on it. So namely what the liver does is it converts Tylenol in the breakdown into various metabolites. And so I'm just going to show that there's a few different kinds, and the key ones are this red one and these blue ones. And I want to highlight that these red metabolites are toxic. And so this NAPQI, when it's produced, actually interacts with other things in your body, namely proteins and nucleic acids, and that damage is toxic and can do lasting and permanent damage to the body. Where there are these other metabolites that are safe, and then these will be slowly interconverted and then eventually excreted through the urinary system. And so this NAPQI can actually be conjugated to be detoxified through this process of GSH conjugation. So this is the normal process. We have our toxic metabolites, and when acetaminophen is normally metabolized in the liver, only about 5% of the acetaminophen is converted into this toxic metabolite, and then it's almost immediately conjugated and neutralized via this GSH conjugation. Alternatively, most of the acetaminophen is converted into these non-toxic metabolites, about 95% of it, and so these aren't going to cause problems for the body, and they can either be excreted directly through the urine or eventually interconverted into other molecules that can also be secreted through the urine. However, if somebody takes too much Tylenol, and it's not actually very much Tylenol, so one of the reasons I'm mentioning this is it's a very cautionary tale because a lot of people take Tylenol and it's very, very easy to overdose on Tylenol. And so what happens if you take too much, then the liver effectively interconverts everything into these non-toxic metabolites, but that consumes all of the reactants. So once those reactants are consumed, these conversions can no longer occur. And so there's less of the Tylenol is converted into these non-toxic metabolites. It can't be conjugated effectively, and that leads to an increase in these toxic metabolites, the NAPQI, which then start reacting with the proteins and the nucleic acids where the Tylenol is being detoxified, specifically in your hepatocytes in your liver. And so as the nucleic acids, the DNA of those cells and the proteins are damaged, it's going to kill those cells and permanently impact them and cause large-term cell death or large-scale cell death in your liver. And so if there is too much Tylenol, it can actually do enough damage that the liver is completely compromised and can no longer effectively do its job. And so I really want to emphasize, like, this is an important thing to keep in mind, especially for children, because if you ever take somebody to the pediatrician, they'll give you very specific dosage instructions based on the child's weight, because it doesn't take too much extra Tylenol to produce these toxic metabolites. And once the damage is done to the liver, it's done to the liver, and that's permanent damage, and it can be fatal. But it also highlights kind of what the liver is capable of. So it's worth noting that Tylenol isn't metabolized into just one thing. The liver can do multiple things with it. So it has lots of different enzymes. It's incredible in terms of the biochemical reactions that it can perform. But there are also potential dangers or problems if you have too much of one thing or the other, because the liver has to deal with these. But it really highlights its role in biochemistry. So Tylenol was a specific example of detoxification, but in normal functioning, in normal physiology, what the liver is really a superhero at is nutrient interconversion in this nutrient pool. So the nutrient pool is really the different nutrients you absorb and your body produces from the digestive system. And it's mainly composed, or it is composed of the organic molecules, the macronutrients, namely the proteins, the carbohydrates, and the lipids or fats. And then nucleic acids kind of fit with our carbohydrates because they have a carbohydrate backbone. And so the key thing I want to highlight here, and the reason I have this really complicated diagram, is it shows how different the liver can basically take different nutrients and it can convert their chemical forms. And so note that proteins and amino acids, when they're broken down, are very different from lipids or carbohydrates in terms of their function in the body. But what the liver can do is it can take these amino acids, it can basically convert them and strip them down into a carbon backbone. And in that process, it can release this NH3, which is eventually converted into urea, because these ammonia compounds can be toxic to the body. But the liver will take these amino acids, strip them down to the carbon backbone, and convert them into pyruvate. And so pyruvate is the product of glycolysis, but glycolysis can also run backwards. So this pyruvate is kind of this keystone molecule that can be interconverted. So the body can also take pyruvate and use that as a carbon backbone to create certain amino acids. Not all of them, but most of them. It can also take that pyruvate to produce glycerol, which is the backbone the backbone for triglycerides, which are kind of the majority of the lipids in your body, or those pyruvates can be used to produce glucose, the sugar that the body uses most commonly in metabolism, and then that glucose can be converted into a larger molecule, a glycogen, which is useful for storage. So effectively what the liver can do is it can take any of these macronutrients that are absorbed, proteins, carbohydrates, and lipids, and interconvert them in between each other, effectively creating the building blocks, the molecular building blocks your cells need in order to function. But it can also take any of these and convert them to pyruvate, which will then feed into the cellular respiration cycle, namely the citric acid cycle and the electron transport chain, to produce ATP. And so in your diet, there are certain nutrients that are necessary that your body can't produce, specifically some essential fatty acids and some essential amino acids. But for the most case, given whatever you eat, your liver can convert things into the form that it needs at the moment. So if you have a low carbohydrate diet, a low sugar diet, that doesn't mean that your body is going to run out of sugars, because it can take the lipids and the proteins and it can create its own sugars using those macromolecules. And specifically for the liver, it's most of the cells in the body can actually break things down into energy, but the liver is really specialized for converting things back and forth. So a lot of the anabolic reactions, or these conversions from amino acids to glucose to fats, really occur in the liver, and that's what it's really good at, because that's not a function that the rest of your body has to do. And then the liver can also monitor blood levels of amino acids and fats and carbohydrates, and then it can be kind of a central regulator for these molecules in the blood and act as a homeostatic organ. So I just want to actually provide these definitions explicitly. Remember that anabolism is the process of building larger molecules from smaller components. So here we have the example of amino acids being combined to produce a larger protein, and this protein will then serve as a machine in the cell, something like an enzyme or a channel, an ion channel or something like that, versus catabolism, which is the breaking down of larger molecules to smaller components. So we take our protein, we break it down into amino acids, and then these amino acids can be further broken down into carbon dioxide and water in order to produce ATP. Finally, because I'm talking about the liver, I'll just mention that it has other functions, specifically storage. So for instance, it can store energy in the form of glycogen. It's also important for phagocytosis. There's immune cells in there. It can break things down. And of course, bioproduction, which we already talked about in the digestive system. So now that we've talked about the liver, I want to shift into specifically discussing metabolic processes. And we'll start with the principles of catabolism, because I think this gives us the core concepts for thinking about how chemicals are interconverted. So as a reminder, when you're breaking down a chemical compound, this involves cleaving or breaking of atomic bonds, and specifically covalent atomic bonds, because ionic bonds can be broken down through charge interactions and things like that. So usually when we think about chemical breakdown, at least in terms of metabolism and specifically organic molecules, we're thinking about breaking these atomic bonds. So here we have a sugar molecule. So this is representing glucose. And so that glucose has 6 carbon atoms. These black circles right here represent the carbons. And then there are also oxygens, and I'm just showing this oxygen here so that you can see the ring-like structure, which is typical of many of the simple sugars. And so combined, this forms a monomer. This would be a glucose molecule or a monomer of a carbohydrate. So here I've just removed the color coding, so now we're just looking at the skeleton. And so each of these solid lines is representing an atomic bond. And as a reminder, that atomic bond is shared electrons between these two molecules. So these two carbon molecules are effectively sharing 2 electrons, 1 from each molecule, and they're sharing them equally. So you can't really identify which carbon they belong to. They're just kind of equally shared. And so each of these atomic bonds is representing 2 electrons that are shared between the molecules that are bonded together, and that's effectively the force that is holding it together. So again, the lines are atomic bonds, and then each of these dots or circles is an atom, whether it's oxygen or carbon. So atomic bonds don't just magically break, at least in normal cases, especially for stable molecules like sugars. And so in order to break these atomic bonds, the body needs to use enzymes, and we already talked about this in the digestive system. And so we'll represent an enzyme, because we're talking about catabolism here, using these scissors. And so these enzymes will effectively break these atomic bonds and free the electron, or cleave the atomic bond and free an electron that is shared between these two atoms. So when an enzyme catalyzes a catabolic reaction and breaks that bond, the shared electrons effectively are released, and so those electrons carry energy and will eventually be bound in ATP. And so one of the things I'll mention here is these carbons have other things bound to them that I'm not showing, specifically in the case of a carbohydrate, water, so oxygens and hydrogens, and I'm simply not showing those. I'm showing mainly the carbon molecules. So the enzyme catalyzes the reaction, it breaks this atomic bond, and in that process, it releases electrons. And these electrons aren't totally free, they're kind of chaperoned, but it frees that electron from being shared between those two atoms. So in the process of breaking down a sugar like glucose, there's lots of atomic bonds. So in order to fully break it down, there need to be multiple catabolic reactions in order to break all of these bonds. So for instance, another enzyme will now cleave this site and release this electron. And so in this case, I'm just showing a cleavage effectively right here, and then this oxygen disappears because we're not showing all of the oxygens that are connected to this carbohydrate. But you can see effectively we've cleaved this 6-carbon molecule now into 2 separate 3-carbon molecules here. And so we're starting to break down from one larger molecule into multiple smaller molecules. So as this process continues, more and more enzymes will catalyze additional reactions, releasing more electrons, but also eventually releasing individual carbon atoms. So as these carbon atoms are cleaved off, this enzyme is chopping off the carbon atoms at the end. These carbons are released, and they're released as carbon dioxide, although I'm not showing the oxygen molecules connected here. We're just tracking the carbons and the electrons. So effectively, we have an enzyme that cleaves these carbons, and it releases them along with the electrons that are holding them together. So through the process of cellular respiration, eventually all of these bonds will be cleaved through a series of chemical reactions. And so what we'll end up with are a bunch of electrons that have been released and a bunch of carbon atoms, which are now kind of by themselves. But remember, these are being carried as carbon dioxide. And here on this slide, I'm just showing explicitly how these carbons are released as carbon dioxide. So now you can see the carbon, and with each of these carbons come 2 oxygen molecules. And as a reminder, these are coming along with the carbohydrate bonds. So these are hydrated carbons, so they're bonded to both hydrogens and oxygens. And so ultimately, through these reactions, the carbons are bonded to oxygens and then released as carbon dioxide, which is a gas, which is then converted into bicarbonate into the blood. So the thing that's really important when we think about cellular respiration, and specifically catabolic reactions that are producing energy, even though that energy is in the form of ATP, and that's often how we think about it, what's really carrying the chemical energy are electrons. And so as a reminder of basic chemistry, when you think about a molecule, and here we have the Bohr model with that nucleus in the middle, and we have our different orbitals. And so as electrons move up and down orbitals, they release or absorb energy. And so those electrons are effectively storing energy in the form of photons. And so every time we see a chemical bond broken, and with it electrons being moved around, there's a change in the energy of the molecule. So when we're thinking about catabolism, we want to track where these electrons are going, because those are going to carry the energy, which is eventually going to power the reaction that then stores the energy in ATP itself. And in fact, it's high energy electrons that are cause the molecule of ATP to be really reactive, which actually power other cellular processes. And so the electrons are what's carrying power, just like they're carrying power in electrical systems. So if we want to see where the energy is going, we simply want to keep track of our electrons. So when electrons are being removed, they create redox reactions. And so you can either reduce a compound or you can oxidize it. So just to help you remember this, reducing a compound refers to the charge. Okay, so electrons have a negative charge. So if we reduce a compound, that effectively means we're adding a negative charge to it, which means it's gaining an electron. So here, if we look at a reaction, if we take a 2-carbon molecule and add another carbon and effectively add an electron here to create a 3-carbon molecule, we've reduced this compound because we've added an electron, which means that this reduced compound has an extra electron, which has carried energy. So this is going to be our higher energy compound, at least in the normal case, especially when we're looking at these cellular respiration reactions. So this means that the reduced compound is going to have an increase in the energy it's storing. And so an example of this would be NAD+. So NAD+ can react with a hydrogen ion, so a proton, and plus 2 electrons. And so when we add these 2 electrons to NAD+ plus H+, we get NADH. So note that the charge balances, 2 electrons, 2 negative charges, 2 positive charges. The NAD is positive, the H+ is positive, and so we get an electrically balanced molecule. But now this NADH has 2 extra electrons, which is extra energy that this molecule is storing. So this is going to be the high energy reduced version of the compound NADH, while NAD+ is going to be the lower energy oxidized version of the compound. This is also true of ATP, or adenosine triphosphate. So this triphosphate or 3 phosphates is really the key thing. Each time we add a phosphate to the ATP molecule, we're effectively concentrating our electrons, yielding a higher energy compound. So here we can see the different reactions. We have AMP, so adenosine monophosphate, 1 phosphate. So here is our pyrophosphate, which is effectively our phosphate right here. And so our AMP just has one of these phosphate groups, and then we react a second one. And so when we add that bond, we're adding a high energy electron. And if we go to ATP, we're adding a 3rd phosphate, again, another electron, which is now extra high energy because these phosphate bonds are very unstable. And so if we look at the ATP, that's the very high energy molecule. And as we break it down, in each of these cases, we'll effectively be releasing an electron, which can then go and power another process. And in that reaction, the ATP loses a phosphate, and ADP itself is still highly energetic and can be converted into AMP to release 2 phosphates, this pyrophosphate. So if we look at the electron carriers, we already mentioned adenosine triphosphate, which is the energy currency of the cell. You've talked a lot about this in general biology. We've talked about it in A&P, but the other ones that we talk less frequently about because they're more specific to cellular respiration are the electron shuttles. And so the first and most, neither one is more important, but the most common one in cellular respiration is this NADH, which we just talked about, which is effectively an electron shuttle. So it's carrying 2 electrons. So by reacting NAD+ plus H+ in these 2 electrons, the NADH is carrying 2 high energy electrons, and it can carry it to other parts of the mitochondrion to pass on that energy to other processes, specifically the electron transport chain, which then powers the movement of protons and oxidative phosphorylation, which ultimately produces ATP. A second electron shuttle, namely a compound whose key function is to move electrons to other reactions and other parts of the cellular respiration chain, is FADH2. And so in this case, we start with FAD. And so we react that FAD with 2 hydrogen ions and 2 electrons. And you can see the hydrogens are added right here. And so by adding these 2 hydrogens, we've added 2 electrons with them. And these are going to be effectively electrons that can be reacted and provide energy for other processes. And so the FADH2 is just carrying these electrons to a different part of the cellular respiration chain, namely the electron transport chain. So that finishes the principles of catabolism that I wanted to cover in this lecture. So as a reminder to review what we talked about, we discussed how food is absorbed in the GI tract and then is processed by the liver, which is really the biochemistry lab of the cell. and it is able to convert the absorbed nutrients into different chemical forms and thereby manage the nutrient pool. So it can either break stuff down through catabolism, so breaking down larger molecules into the smaller molecules, which we have here a glucose molecule being eventually broken down into energy in the form of electrons and carbon dioxide molecules. Or it can also build up molecules, smaller molecules, into larger molecules, which we see here in our nutrient pool, where the liver can take glucose and basically bond those glucoses together to form glycogen. It can take amino acids and build proteins, or it can build lipids of different kinds by combining glycerol along with fatty acids. So this nutrient pool is really important. It's largely managed by the liver, but basically most of the cells in the body are capable of catabolizing glucose, and basically all of the cells in your body do some form of catabolic reaction. The next part of the lecture, we really talked about how to track energy through the process of catabolism and how energy is stored in chemical bonds and electrons. And so if we can track where electrons are moving when chemical bonds are broken, we can effectively track where the energy is going. And specifically, there are shuttle molecules in these different cycles or processes in cellular respiration, specifically glycolysis and the citric acid cycle and this preparatory step in between, where electrons or chemical bonds are broken, releasing electrons, which are then captured by shuttle molecules and moved to different parts of the pathway. And so that's really represented by these yellow lines here in this pathway, where all of these shuttle molecules are taking the electrons and eventually bringing them to the appropriately named electron transport chain. And then finally, we talked about, in general, the process by which organic macromolecules are broken down into CO2 via the action of enzymes, which are breaking these chemical bonds and releasing the electrons to be used for energy production. So I hope that's helpful. This provides a general overview. And so when you're thinking about metabolism, it's always useful to think about why these different molecules are important, where are the electrons going? And I'll just mention, in the breaking of these bonds, often these bonds, when they're broken, I'm showing these electrons going off, but they'll also often go with hydrogen. So this molecule is being reorganized in more complicated ways that I'm not showing. This is just a very simplified diagram sketching out the process and how to think about it, where we go from a solid sugar here into gas and energy.